All right, now we'll look to our last talk that Dr. Siewert will give and talk about the interesting study 1100 in combination with checkpoint inhibitors. After his talk, we'll have a 15-minute break, and then everyone can come back, and we'll have a half-hour panel discussion with everyone, plus Dr. Weiss. So, thanks.
Yeah, thank you, Jeff, and thank you to Nanobiotix for inviting me. I always love going to New York because I can fly into LaGuardia, and because there is always a delay, I can stay longer home. So I had an extra 2 hours with my family yesterday, so that's a nice thing. I do worry that maybe in 10 years they will have a new airport and that might go away. With that being said, the other comment I would like to make is, rarely I have a panel of 3 radiation oncologists and 1 medical oncologist. That seems a little bit out of balance, and we really make progress while they give the same darn radiation all the time. But it sounds like maybe with a nanoparticle that might change, and so I think that's the reason why, arguably I'm so excited about this. I'll show you this here. These are, I guess, my disclaimers. I wanted to put a picture of Jose Bazalga here, especially in New York. But then I thought maybe it will give me the wrong impression. But these are my disclosures. So just to tell you a brief story, and I think Jared said it really nicely in one of his comments. Our time is valuable. We don't do things that are not likely to lead to success, at least. We make estimates, we make guesses. We think this is valuable and it's going to have a big impact. That's why we never got involved in a radioprotectant trial. We saw the ASCO data and we thought, that's interesting. It'll take a lot to make the standard of care. And so and there were other challenges also logistically, but we didn't do this. But what happened? How did I get involved in this? I'll tell you the story. There was a meeting in Barcelona, a head and neck European head and neck meeting called ICNO. And in the hallway, 2 folks came up to me and say, "Hey, we've got this really interesting stuff. You want to take a look?" And I'm like, "Okay." And Bob Ferris was also there, actually, and it was Nanobiotix. And they said, "We've got this cool new technology. It's a nanoparticle." And I'm like, "Well, nanoparticle. Everybody has nanoparticles." And I was very, thinking, "Is this really worth my time?" And then in the hallway, literally within like 10 minutes, they showed me a lot of data. But there was this one slide where I started to think, there are these. So they showed me this one slide or this one data set that not only did they kill tumors because they have more radiation, but there was this whopping interferon gamma response. And people who know me, I've talked a lot about interferon gamma, and I think you just heard Dr. Welch's talk about interferon gamma response. That's the highlight of an immune response. And not only was there a whopping interferon gamma response, but it was in sarcomas, which are dead, cold, desert, immune desert type tumors. And that's when I started listening. And it's nice, I think it's now a year and a half later, to stand here and actually talk about a trial that we designed specifically around the idea of making cold tumors hot. It's a term that's arguably overused, and I would be a little careful, but in principle it's true. If you can make a cold tumor hot, everything is going to work better, whether it's radiation or chemotherapy or immunotherapy. And at the time in Barcelona, they asked me, "What do you think we should do? This is very different from what everybody else is doing. What should we do as a proof of concept to convince, everybody, convince the world that this is worthwhile?" And I said, if you really want to convince people that this is different and better, you have to go into a really tough setting. You have to show that it works somewhere where arguably nothing else really works. And I proposed 2 things. I said, one is go into patients who fail immunotherapy, who are checkpoint failures, where arguably the bar is very high and we don't have good treatments. And then from my personal perspective, I treat a lot of head and neck cancer, but it also applies, to pancreatic cancer, lung cancer, radio-resistance patients who have failed prior radiation. For head and neck cancer, these patients do terrible. Even though they have a localized disease, in principle they should be curable, it's localized. We can't do it because the radiation no longer works and they're not resectable. And so I said, if you look at these 2 populations, and you show activity, that would convince everybody in the field very quickly, even with a very small sample set. And that's really what this trial is all about. That's what the 1100 trial is about. I mean, I was expecting them to say, no, that's too tough, we don't want to do it. But they said, we believe in this agent, and we're going to do this. And then we followed up at ASCO last year, and then this year. And I think this trial is about to launch maybe in a month or so, or 2 months. It's very close. And I'll talk to you briefly about this trial. But that's the story behind this. I mean, I do a lot of things that are iterative, slightly better, chemotherapy, slightly better radiation. But once in a while, you have to do something that's maybe a bit higher risk that has potential to revolutionize things or really change things. This is, disruptive potentially. And, you have to take it with a grain of salt. There's risk involved in that. And when I was a fellow, I did an oncolytic virus, maybe 10 years before. It was cool to do oncolytic virus. It was interesting. We learned something. It didn't work. However, now oncolytic viruses are again in vogue. So this is a trial that's designed to potentially be disruptive. That's the idea behind it. That's the 1100 trial. So the patient population is very similar to what I outlined in the original meeting in Barcelona. Take patients who have failed prior radiation, who are radiorefractory, who are not likely to be cured, who are, even though they have localized disease, they are considered incurable. And then take patients who are checkpoint inhibitor refractory, and those are the two populations. We initially said we would do this for head and neck cancer, but then we said, the biology is so similar between head and neck and lung cancer, why not do it in both? And so we have a lung cancer cohort as well, and obviously it makes it more applicable. But in principle, make no mistake, if this works, there's no reason to apply this to other radiorefractory cancers or immune-resistant cancers, bladder cancer, mesothelioma, pancreatic cancer, breast cancer. If it works, once you have a proof of principle, it's easy then to put it into other settings. So it's right now a trial, a phase 1/2 trial, and right now we have 3 centers, Chicago, UNC, and Jared said, I convinced him with the systemic effect, and I'm glad of this. And then we have a center in Atlanta, and there may be other centers joining. But I've actually been very happy, when I talk to people realize this is different and has potential. To give you the background, obviously, in head and neck cancer right now, we have approval of 2 PD-1 agents. We have Pembro and Nivo approved. In lung cancer, we also have atezolizumab and durvalumab. But in principle, they work in similar settings. So this is right now the land of the state of the field. We have approval. And just to show you how this works and give you another example, I always show this case of an immune-responsive cancer. This is one of my patients treated on one of the keynote studies, actually. Very large base of floor of the mouth tumor, treated with 2 rounds of radiation, clearly not amenable to any more radiation, refractory to multiple lines of chemotherapy, not a surgical candidate, a lot of skin lesions. But what this case really highlighted, and this patient was sent to me saying maybe this patient should go to hospice. The patient was enrolled on a study with pembrolizumab, and within about a week, and I've never seen a response that was so fast, the skin lesions on the side of his neck started to show the central necrosis. And then within about 3 weeks, many of them were gone. And within about 5 or 6 weeks, the entire tumor was gone. This is an unusually fast response. This is not usually how large tumors disappear. Usually it takes a lot longer, up to 12 or 20 weeks. And we actually don't know. Why did this cancer respond so incredibly well? Maybe there's a lot of mutations, a lot of antigens, maybe a very active immune response. But we really don't know. We don't understand why sometimes these immune responses are much faster. But it goes back to the underlying biology that I think was already introduced by Dr. Welch. There's 2 things that we believe drive immune responses. One is the underlying foreignness of the tumor, and that could be primarily mutations. If you have more mutations, there's more chances for the immune system to recognize a tumor, more antigenic. It could also be a virus, and it's a bit playing the lottery. You might have the right mutation even though you have a low tumor mutational burden, but overall in a large population of patients, the more mutations you have, it will correlate with benefit. And that's upstream. But when you go downstream, what you really need is inflammation, and that's where the gamma interferon responses come in. The T cells here, gamma interferon right here, which leads to PD-L1 expression, essentially influx of CD4s and CD8s, and an inhibited immune response that then you can reverse with a checkpoint inhibitor. That's how it's supposed to be. So these two are currently the most prominent biomarkers: tumor mutational burden and then inflammation, and you can read this out by either PD-L1 or maybe some signature of interferon gamma, but that's how we think it works right now. And this is an example of showing you this maybe a little bit better. This is a tumor that's heavily infiltrated. Green here are the CD8 cells. Red is a tumor. And this is a highly inflamed tumor. On the other hand, this is an immune desert tumor. This is how a tumor looks that has no immune response. And so the obvious challenge is, this is obviously a tumor where we have to generate immune response for the immune system to work. How do you turn a cold tumor into one of these more inflamed tumors? However, and this is a simple slide, this is what people always talk about, hot tumors, cold tumors, how do you turn this into this? But it's actually a little bit more complicated. So this is where checkpoint inhibitors are supposed to work. And maybe they work 30, 40% of the time, but the majority still don't work. Still in this setting, checkpoint inhibitors don't work. And then makes things even more complicated. Even if the checkpoint inhibitor worked here, I would say at least 2/3 of the time after having a response, there will be the emergence of resistance at some point. So even in the population where we already have a good system, we are still not anywhere close to where we want to be with checkpoint inhibitors. And that's arguably the low-hanging fruit. This is the holy grail. How do you turn cold tumors hot? And that's where I think injectable agents may potentially play a role. And I think doing nanoparticles and using radiation is maybe one of the most promising strategies to do that. But sting agents and other things are also being explored in this setting. And just to show you, this is a slide from Ira Melman / Dan Chen that actually show that it's actually even a little bit more complicated. So inflamed here, I showed you a picture, immune desert, but then there's also kind of this excluded phenotype. So the tumor is immunogenic, generates the right immune response, but has walls up that the immune cells can't make it in. And we think this might be related to TGF-beta and some other factors like angiogenesis. But when you think about all the recent data that suggests chemotherapy plus a checkpoint inhibitor works better. Look at all the great lung data, and we just heard some updated data from World Lung, squamous, small cell, adenocarcinomas. Or think about Pacific, you give radiation, you kill tumor. So I think there's something about killing tumors that breaks down the defenses of the tumor, that kind of makes the tumor in disarray. And that's likely why when you combine a traditional cytotoxic agent, whether it's radiation or chemotherapy or something else, with an immunotherapy, why it works so well, because rather than being able to maintain that immune-excluded phenotype, likely you are highly, highly disruptive. So there's something good about killing tumors up front. And again, I think having the idea of just injecting an agent in the middle might not be sufficient, but if you radiate on top of it, you might actually really disrupt the tumor and put it in so much disarray that an immune response might be much more effective. So now let me talk specifically about head and neck cancer and provide you the background to where we are. This is currently the lay of the land with trials. We have approval for second line, so after platinum failure, of either pembrolizumab or nivolumab. At ESMO, we'll see data on KEYNOTE-48 that I think will be quite impressive. I cannot say more than that. Single agent and chemotherapy, essentially recapitulating what I think we have for lung cancer. But make no mistakes, these are metastatic disease patients, and even if these drugs work, these survival curves still fall to like 30-40%. The majority of patients will die either without a response or even if they have a response, become resistant. There are efforts to go into the locally advanced setting. There are efforts neoadjuvantly, and there are efforts in the adjuvant setting. This is Pacific-like. But in principle, while this is a crowded space, none of these phase 3 studies, these large studies, addresses PD-1 failures. And that's really, I think, the the setting where it's very hard to actually find traction, to get traction. There are early trials, but that's what we're going to look at with the 1100 trial. Somebody fails after Keynote 48 treatment, after nivolumab, or even after a Pacific-like setting. That's where potentially a treatment approach that overcomes immunotherapy resistance, or potentially in the curative intent setting, radioresistance, really makes sense. So these are the two settings that are already introduced at the beginning. Patients who failed prior curative intent setting, who are truly radioresistant, these patients would typically be treated with a PD-1 inhibitor. Median time to progression is usually short and survival isn't very good. Or PD-1 failure patients who have failed a prior checkpoint inhibitor, just going back to the trials that I just showed you. And this is now reflected in this trial. So the 1100 trial has 3 cohorts, and I'll walk you through here. So cohort 1 is localized disease, and this is shown here in this picture. So patients, and we have 2 subgroups, group A and group B. Group A is essentially patients who have failed prior radiation, who would be amenable to re-irradiation. So they have localized disease, they're considered radiorefractory, but they have not been pretreated with systemic agents, but they have failed prior radiation. So this is truly radiorefractory. Group B will test a little bit more the idea of, can we have a systemic effect in addition? So these are patients who are amenable to re-radiation, but they also have metastatic disease at the same time. So kind of this reflects the 2 types of failure patterns that we see, local failure or local failure plus distant failure. And then cohorts 2 and 3 focus just on distant disease. Cohort 2 is aimed at lung metastases. Lung metastases are extremely common, obviously. And then cohort 3 is liver metastases. As I mentioned before, while this was originally decided to or developed for head and neck cancer, we said there's so much overlap with lung cancer, and it's a big, potentially a big source of patients, and biology should be similar. So this trial does include both head and neck cancers and also non-small cell lung cancer patients. Any questions right now about the 3 cohorts? That's the idea behind the trial, is to take the toughest patients, radiation resistant and checkpoint inhibitor failure patients in these cohorts. And just briefly to walk you through, the study has 2 parts. Probably the bigger part is the dose escalation part, where we have 3 dose levels. So R3 is given at a percentage of 15% with hypofractionated radiation. I remember discussing this with one of our radiation oncologists, James Malatek. How should we do the radiation? Going back to the prior discussion, we said we should do hypofractionated radiation because it is the best way of generating immune response. In the end, all we really want is to make these tumors more immunogenic, make these tumors hot, and then have the concurrent checkpoint inhibitor work. And so this study has concurrent checkpoint inhibitor with R3, and radiation is an activator to R3. So initially 15%, then 22%, and then we actually go up on the radiation dose somewhat from 40 grays overall to 45 grays. And then there will be an expansion cohort. You can see here the details here, 40, 40, 45 gray. Cohort 2 and 3, these are lung and liver lesions, trying to cover typical metastatic sites and give appropriate radiation doses. Here we have 45 gray, 50. Again, a dose expansion, 15, 22, 22, and going up from 9 times 5 to 10 times 5. And then this is the liver cohort, 13 gray, and going up from 15 to 22%. So this is a trial design, and we will give a concurrent PD-1 inhibitor. We actually made it open to either one, either Nivo or Pembro, which would be available commercially, as this is an indication that's FDA approved. Just to show you the trial outline, and I don't want to bore you too much, but this is how these schemas usually run. So you have the patient enrolled on the study. You have about a 4-week window to starting treatment. Then we will start with the radiation doses. Here, the administration of R3, and then radiation starts with the typical fractionation. Here are the 3 cohorts. And then we continue with the immune checkpoint inhibitors the entire period afterwards, after radiation. And then we have follow-up scans here. This is phase 1, phase 2 part, the dose escalation part. And then should go further. This is, the expansion cohort where once we have a dose, we'll actually enroll more patients to get a better signal of activity. And this is where we'll look primarily then at response rate as an outcome measure. So that's a trial in principle. And just kind of, again, trying to give you a bit the bigger picture, which is, this is a graph that one of my colleagues actually put up, Ezra Cohen, highlighting, where we are in oncology. Curability versus creativity. And there's an inverse relationship. When you have highly incurable patients with recurrent metastatic disease, you can be very creative. And that's why we're starting here, obviously, with R3 in this trial setting, patients who are incurable. But make no mistake, this is an agent that potentially synergizes perfectly with our curative intent options, which is largely radiation-based. So, moving from here to here isn't that difficult. In fact, in our trial, what we're already trying to do is we take patients who are just incurable because they have localized disease that's radio-resistant, but in principle could be curable. So we're already moving in this direction. This is really the large prize, the grand prize. And the pharma company knows this. So that's why in the previous slide I showed you, there's all these local region advanced trials that are doing chemoradiation plus pembrolizumab plus nivolumab plus another PD-L1 agent. However, I would argue, and going back to Dr. Welch's talk, doing regular chemoradiation, regular fractionated 70 gray over 7 weeks is highly immunosuppressive. And I'm a little bit worried that might not be the right approach. So in addition, with our 1100 trial, trying to get at this population, I also think we have the opportunity, if it works well, to potentially go into the advanced setting where I think there's a lot of things that are possible. And then obviously, finally, we have early stage, and if you can give a lot lower radiation doses, maybe that would be optional, but that's really far away, so I'll leave this just more as a future. So let me summarize. So head and neck cancer and arguably lung cancer are obviously great diseases for immune checkpoint inhibitors, but still there's a large unmet need because checkpoint inhibitor failures are either constitutive up front, they don't respond, or even if they do respond, there's a large population of patients who become resistant, who have acquired resistance. So constitutive and acquired resistance is a big challenge. Checkpoint inhibitor resistance. The other big challenge in our field is radioresistance, and that's arguably across oncology. We don't use radiation for some tumors because these tumors are just not amenable to radiation. If you can potentially, ninefold increase radiation, maybe that becomes a radiosensitive disease. And then importantly, in addition to these two indications, making tumors radiosensitive again may also induce immune response, and that's really where I got interested. And that's where the 1100 trial really comes in these two populations, checkpoint inhibitor resistant, radio-resistant, and then looking at these populations and trying to overcome both immunotherapy resistance and radio-resistance. That's all I have. Thank you so much for your attention, and I'll be happy to take some questions. I can make one more comment that I forgot to make. So I'm a bit of a skeptic about intratumoral agent, or at least I used to be, because it takes effort. It's not something that I would typically do, and I've done my fair share of intratumoral agents. Be it the oncolytic virus, or we did a plasmid recently. You have to have a large effect size for physicians to really use it. So it is not something that you would do if it's just slightly better. And thinking about other tumor types, if you have to go by endoscopy or so. So in my mind, if you can really make a large difference for a population, then it's worth it. Arguably, I would say, you can see the struggle that TVEC is having with melanoma, even though it's indicated. So I think the fact that this is given, R3 is given a single time, which makes it a lot easier. If you do it repeatedly, it's challenging. But giving a single time and these really massive effect size that I think we at least preliminarily see, that's in my mind makes it worthwhile and potentially could change the field. So I'll add this on, that intratumoral agents are not simple, and there's whole ad boards where they talk for 2 days about how to make this feasible, because it's not something that usually we do. So changing standard of practice, just even logistically, can be challenging. make everybody fall asleep? No.
I think maybe everyone needs
Everybody
a break.
wants to go to lunch.
I'm sure there'll be more questions when we're at the panel after everyone has a chance to have coffee and a bio break. So, no questions? All right, let's take a 15-minute break then. Thank you, Dr. Siewert.
Thank you.